Biotechnology and stem cells. Where to start? Well, first, you have to know what stem cells are. Technically, stem cells are cells that have yet to differentiate into their final form. To find out more, we must first explore what types of stem cells there are. Firstly, they can be classified by their plasticity, which means how much they can differentiate. Pluripotent cells can become any cell in the body, as well as the cells required to make another organism. They come from the first few divisions after a zygote is created. Pluripotent cells, on the other hand, can become every cell in the body except the cells of the placenta, which means that it cannot create another organism. These come after a few more divisions when the blastocyst is created. Multipotent cells are more specialized and can only differentiate into a few cells. Oligopotent cells are similar to multipotent cells, except that they have even fewer specializations. Unipotent cells can only replicate themselves. Stem cells can also be classified by their source. Adult stem cells can be either unipotent or multipotent, and embryonic stem cells can be either totipotent, pluripotent, or multipotent. Now that we know what they are, how are they being used? Well, adult stem cells have been used for a while, and how is best explained in an example. In a bone marrow transplant, someone whose bone marrow is healthy has their bone marrow used to replace someone's whose bone marrow is not producing blood cells correctly. This happens in leukemia and can help fight the disease. Now, like everything, there are pros and cons with adult stem cell technology. The main pro is that it is tested to work for what it does, and does that well. The cons are that if the match is not perfect, there's a chance of rejection, and that only a few types of cells are regenerated from adult stem cells. So for example, nerve cells cannot be regenerated through adult stem cells. Embryonic stem cell transplants solve this issue. In these, the tissue has to be acquired from the inner part of a blastocyst. This is then grown in a culture, being split periodically and being provided feeder cells and a serum so that it can not specialize and it has nutrients to grow. These cells are then placed where the damaged tissue is and then can be allowed to differentiate into the tissue which needs replacing. The pros are that any type of cell can be created from an embryonic stem cell, and the cons are that there is a chance of rejection still, and it is higher than adult stem cells because it's harder to find a perfect match, and the technology is still in a very experimental stage. Here are some examples of embryonic stem cells at work.
Also hopeful, California researcher Hans Kierstead.
It's going to result in a floodgate of more researchers, more students entering the field, and greater industry investment.
He's experimented on paralyzed rats, injecting them with embryonic stem cells to generate new nerve cells. The rats walked again.
Think about these sorts of diseases, you have to think about this idea that these stem cells are going to go in there and fix a discrete problem. So problems like diabetes, for example, where the pancreas is not making enough insulin. Problems like Parkinson's disease, where you're simply not making enough dopamine in the brain. Those are the types of diseases, those discrete diseases that are probably going to be the most responsive. The same thing with spinal cord injury.
So what if you want the best of both worlds? Well, this is where induced pluripotent stem cells come in. See, all cells in an organism have the same DNA. It is the transcription factors that decide what parts are used for creating proteins, and thus decide what the cell does. Figuring this out, scientists have forced embryonic transcription factors onto skin cells and therefore have induced pluripotent traits onto non-pluripotent cells. This means that they have the cells with the pros of both, and as you can use regular old skin cells from the person who has the issue, the chance of rejection is pretty much killed. The only issue is that it is still very experimental, and more research is needed to solve any issues that may arise. Here are some examples of induced pluripotent stem cells at work.
Like these beating heart cells that started out as facial skin.
We can take a skin cell from any human and make a stem cell line from that human. That opens the door to therapy.
Doctors can take the skin cells from someone with Alzheimer's, turn them into nerve cells, and try different drugs in the petri dish without experimenting on the patient.
These are methods that can now be used by laboratories all over the world, and I guarantee you, dozens if not hundreds of scientists will be jumping on them.
Now that you know the results of this treatment, it is important to give credit to the researchers. The first successful stem cell transplant occurred when Edward Donald Thomas used bone marrow treatment for leukemia in 1956. This opened the door for any other types of stem cell treatments. The next two massive breakthroughs were made by the same person, James Thomson, who isolated the first human embryonic stem cells in 1998, proving that it was possible and allowing for a whole new outlook to be created. Then, in 2007, Thomson again made a breakthrough with the first induced pluripotent human stem cells. These two breakthroughs combined mean that stem cells are now accessible to scientists and researchers who want to experiment with them. This accessibility means that research can be improved even further and more treatments can be created. This induced pluripotent technology might reduce the ethical concerns that some had with stem cells in the past. These ethical concerns are a main reason that progress has been so slow on improving treatments. Now, however, things are changing, and this map shows that most major research areas, including Canada and the United States, allow for stem cell research and for government funding of stem cell research, which is necessary for future progress.